Hey everyone, my name is Jessica Dela Cruz. It is February 13, 2019. It is Bio 225. This is week 6, environmental influences on microbial growth, salt tolerance testing. It is exercise 2. So this, and what we're going to be doing is we are here on my notes. So we're going to actually be preparing our pH broths here. I have pH 5, pH 7, and pH 9. I've already done my S. epidermidis broths there. So in this lab, I'm going to show you with my S. cerevisiae. Okay, so what I'm going to do is I'm going to get my active culture broth. Actually, I'm going to grab my pH 5 first. I'm going to open this. And I'm going to pass it over the flame. Okay. And so with the pH 5, I am going to add 1 drop of the hydrochloric acid. So just 1 drop to adjust the level of the pH. Okay. And then flame that. Flame the lid to sterilize. And cap that. Okay. I'm going to skip over the 7 because the 7 is already adjusted to its pH. So no acid or base is needed. So I'm going to grab my pH 9. I'm going to pass it over the flame to sterilize. And I'm going to put my sodium hydroxide. I'm going to put 1 drop of this in here. Okay, perfect. Pass that over the flame to sterilize. Alright, and now what I'm going to do is I'm going to grab my active culture broth, or I'm going to grab my pH broth, and I'm going to get my active culture broth, and I'm going to put 2 drops of my active culture broth into my pH, into the pH 5, 7, and 9. Okay, so I'm going to grab I have my active pH 5 broth, so I'm going to flame that to sterilize. Grab my active culture S. cerevisiae. Going to pass that over the flame too. Okay. I'm going to grab my pipette that's in alcohol. Make sure it's all dry. Alright, now I'm going to grab 2 drops of my active culture broth. Okay. I'm going to put that 2 drops into my pH 5 broth. 1, 2. Okay. So I'm going to flame my active culture broth to sterilize it. And I'm going to recap it. Do the same thing for my pH 5 broth. Okay, I'm going to grab my pH 7. Pass that over the flame. And I'm going to grab my active culture S. cerevisiae. Pass that over the flame as well. And I'm going to grab 2 drops again. I'm going to put the 2 drops into my pH 7 broth. 1, 2. Oh, perfect. Okay. I'm going to flame the lip of my active culture broth. Like that. Flame my pH 7 broth. Like that. Okay. Grab my pH 9 now. Pass that over the flame to sterilize. And then once again my S. cerevisiae. Pass that over the flame to sterilize. Okay. And then now, I'm going to grab 2 drops again. And put that into my pH 9 broth. 1, 2. Okay. And now that I'm done with this, I can dispose the pipette into my 10% bleach. Pipette, or not pipette. Flame my active culture broth to sterilize. Do the same thing for my pH 9 broth. Okay, now that I've inoculated these broths, what I'm going to do is I am going to take a picture of them. Now that I have putted the active culture broths into my pH broths, and then I'm going to incubate these for 24 hours in my incubation location. And I'm going to, after the 24 hours, I'm going to observe for growth, which shall appear as turbidity. If no growth is observed, I'm going to incubate it for another 24 hours. And then from there, when I see growth, I'm also going to take a picture to just compare and contrast. Alright, and that's it for this lab. Bye!